Welcome to Cell Culture Community, where we explore how bioengineering can transform human and planetary health. Today, we discuss a new class of diseases, not infectious or driven by aging and lifestyle, but rather a genetic blood disorder. Sickle cell disease may not seem connected to health access, but its burden is concentrated in sub-Saharan Africa, tribal parts of India, and the Middle East. So, patient outcomes are heavily shaped by global inequities and biases. The solution we look at today is a refreshing example of how accessibility and impact can be pursued in a high-income country, within the parameters of the for-profit market. Before we jump in, this is the last episode of this season of Cell Culture Community. If you've enjoyed your time with us, let me know by rating the podcast or sharing it with a colleague or friend. If you're new here, check out the first 6 episodes for more inspiring stories of science and technology solving global health challenges. I'm with John Paderi, Chief Scientific Officer at IHP Therapeutics. He's developing a cutting-edge therapy for sickle cell pain crisis. We'll talk about what that is in a second, but their self-administered therapy will empower patients by shifting care into the home setting, avoiding costly and often ineffective hospital visits. So, John, sickle cell pain crisis is not something I was super familiar with before researching on it for this podcast. So, to kick us off, could you explain what the condition is and what pain crisis in specific is, which is what IHP is going after?
Absolutely, and thanks for having me. So, sickle cell disease is a genetic disease. It's something that you acquire from your parents. And it's a disease of red blood cells, specifically the hemoglobin in your red blood cells. It changes the shape of the red blood cell and makes it both fragile, but it also makes it what we call sticky. In other words, it blocks blood vessels. It'll stick to the vessel wall, and the downstream organs and tissues like your bones and your kidneys, liver, lungs, when these are not getting the blood supply, then those go through what it's a ischemia, or the lack of a blood supply causes injury, and that is in itself a very painful episode. And those are what we call pain crises. It's really the hallmark of sickle cell disease.
And they're called pain crises, but they also have an impact on organ functioning in the long run. Is that right?
That's correct. Yeah. So it's not just a disease that is painful. What's underlying that pain is actually these pathomechanisms of the organs are not getting blood supply. So you'll oftentimes see individuals with sickle cell who have kidney disease at a much earlier stage than would be normal. You can have a condition called acute chest syndrome. This is when you're in a vaso-occlusive episode, and that again is caused by this lack of blood flow that's feeding the organs.
What is the burden for sickle cell disease like around the world in different populations? And I came across some statistics that the burden is actually growing. If that is the case, why so?
Yeah, so sickle cell, there's 100,000 people in the United States who have sickle cell disease. And there's more than 7.5 million people worldwide. This is something that, it's a lot more prevalent than we think of. It's considered a rare disease in the United States. But there's quite a few people who are afflicted, and part of the reason is that the sickle trait is, so if you only have one of the genes, that is actually protective from malaria. So malaria is a parasite that propagates within your red blood cells, and if you have one of those genes, that actually makes it a bit more inhospitable for the parasite to propagate. So anywhere there's malaria, you're going to see sickle cell disease as well.
The sickle cell trait is common in tropical areas with high malaria incidence. Its prevalence has been growing due to higher birth rates among these populations. How has it stuck around in the US for so long? We don't hear about malaria as a public health crisis in the West anymore.
Right, right. Well, I mean, for all the reasons that we have folks from all around the world in the United States, some by choice and obviously some not, that historically have reside in the United States today, and that's kind of how we have the condition here. sickle trait is not in and of itself very dangerous. People live a normal life. So you might not know that you have this gene that you could be passing along.
Next, we understand patients' experiences with sickle cell pain crisis. Some surprising similarities to migraines. They are unpredictable, triggered by different kinds of stress, and often preceded by an aura that the patient can intuit. Let's jump into pain crisis itself a little bit. What causes pain crisis, and how does it kind of feel for the patient? How does it look for the people around them?
So a pain crisis can come on for a variety of reasons. Some folks know what will trigger an event, and this could be dehydration in hot climates. It could be actually cold. That also is one of the triggers. So folks taking a cold swim is oftentimes a trigger, and it could also then be a cold. You get the flu, and somebody without sickle cell disease is just going to have other general inflammation, but that inflammation can also cause a vaso-occlusive event.
In a vaso-occlusive event, sickled red blood cells block blood flow, depriving tissues of oxygen.
Now, the vaso-occlusive events can look a little different for every individual. Frequently, there are preceding symptoms, and they're oftentimes called prodromal symptoms, and this could be maybe a milder version of what a full-blown, what we call pain crisis would look like, or it could be different symptoms like a tingling fingers or an achy back. Something that alerts you this is coming on. You can think of it in a lot of ways like a migraine that has aura. The aura is not a migraine, but it's that kind of preceding symptom. Once you get into a pain crisis, the full-blown pain crisis, it's just debilitatingly painful. Right now, the only thing we have for managing pain are opioids, and it's oftentimes a much higher dose of opioids, depending on how long you've suffered from sickle cell disease, how many times you have these events. And that's only masking the pain. That's actually not doing anything for the underlying pathophysiology. I can tell you, many, many years ago, I was a camp counselor at a summer camp that had, we were taking care of a cabin of kids who had sickle cell disease. And after a cold swim, a young boy, he came up to me and said, "Hey, I'm not feeling well." And I said, "Well, he's somewhere around 6 years old at the time. Are you not feeling well? You're homesick? Or are you not feeling well? It's the flu." And he said, "No, this is sickle cell." So even at a very young age, he knew what symptoms of sickle cell were. And I can say that, he deteriorated very quickly and just writhing in pain. It is just a really horrific thing to see, and especially at such a young age.
Yeah, that's terrible. And I guess because it's genetic, everyone's experiencing it from an extremely early age. That's just kind of how the disease works.
That's right. Yeah. You're definitely, young children kind of going through that sort of pain that, hopefully nobody at any age really should experience. It's really hard to see.
And you made the migraine comparison. Is it similar to migraines, kind of difficult to predict, pinpoint accurately, treat the root cause for pain crisis? Does that analogy carry over?
So it's a great question. What's nice about migraines, if there is something nice about migraines, is that we do actually have some medications that work. There are drugs called triptans that you will take at the sort of earliest onset. And in many cases, that can actually prevent the full-blown migraine from occurring. Right now in sickle cell disease, in some parts of the world and parts of the U.S., there are what we have called day hospitals that you can go, and hydration and early treatment of the symptoms with pain medications. Those are kind of what would be most common. Make sure you're getting rest, these sorts of things, because lack of sleep can also then further exacerbate the pain crisis. So we do have those, but I would say today is a lot more challenging. We don't have the triptan equivalent for addressing the underlying pathophysiology.
Today, the best treatment for severe pain crisis is opioids, which come with racial stigmas around drug use, have side effects, and don't treat the root cause. We explore the therapeutic landscape to identify the gap IHP aims to fill. It sounds hard. You were saying earlier that most people end up getting treated with opioids and the doses go up as you go older. I was reading online about cyclic withdrawal symptoms as patients get on and off opioids. And there's a whole set of challenges around, what's the person's relationship with the drug? How are they able to manage that? How do doctors perceive that relationship? And so on and so forth.
Yeah, so that gets even more complicated. Opioids are really the only medication that we have that masks pain during one of these vaso-occlusive episodes. Obviously, opioids have their, they are addictive. They have a lot of issues with them in that they don't work as well. The more you use them, they can cause what's called chronic pain syndrome. And this would be more central, kind of in the central nervous system that gives you like a phantom pain or a very like a hypersensitivity to different stimulus. And then what I was saying is much worse than that is that an individual comes into a hospital and they're in pain, but you can't measure pain. You just ask the individual what they're feeling. And they know because of multiple previous vaso-occlusive episodes, I need to have this dose of whatever that opioid would be. And now all of a sudden they're treated as a drug seeker. So this is, a dangerous and utterly debilitating situation. And then the healthcare system is saying, well, we don't believe you, and we're going to not treat you with respect that you would deserve. And there are racial stigmas associated with this. Unfortunately, this isn't anecdotal. This is very well documented by the NIH, the FDA, et cetera. It's really just a terrible situation for individuals with sickle cell.
Yeah. And I can imagine it's just that much harder to seek care if you aren't on a formal insurance or don't have the same level of documentation of your disease as someone else might. Yeah.
I think that's right. The day hospitals in areas where there are kind of sickle cell centers of excellence, those are really well suited. And I would say if you're close to your hematologist, sometimes the hematologist can help facilitate the better care. But otherwise, you're sort of entering the emergency department and you're kind of with everybody else. And the staff there are typically not going to be trained for sickle cell and how to manage this. And so it really does become a major concern. And part of this then goes into, well, if the last time I went to the hospital, I was treated so poorly, why would I go back? And, it's not that this is just pain that you can manage at home. There are also, dangerous complications that can occur. So really, it's just a wholly inadequate situation that we're in right now.
That makes sense. And we'll get soon to the therapy your company's working on, which will hopefully change the situation. But I'm also curious when we think about why it's been so difficult to treat and develop a kind of root cause cure to this so far. Sounds like part of it is maybe could be racial biases, could be a very scientifically difficult problem. I'm sure you know the science of this area very well. What do you feel has made it so that it has remained untreated for so long?
Yeah, it's a great question. And, you can't answer these things with 100% certainty, but it is pretty striking if you look at, in the last decade, how much money has been spent on smaller diseases or diseases that affect far fewer individuals. Let's just take cystic fibrosis. I think is kind of the classic comparator. There's about a third the number of people in the United States who have cystic fibrosis versus sickle cell disease. And there's a lot of parallels there as far as life expectancy goes. And despite there being a third the number of individuals, there's largely over 3 times the amount of money that has gone into cystic fibrosis therapies as opposed to sickle cell. But that's not to dismiss the fact that it's complicated. And, so really understanding the pathomechanisms of a vaso-occlusive event, we're getting better. And I think that we've really made some leaps in understanding what's going on and then how to address that. I would also say from a funding perspective, at least government agencies have really increased their awareness and are really encouraging folks to put some effort into sickle cell and figuring out how to, right now, aside from gene therapies, there are only 4 drugs that are approved for sickle cell today. And primarily one, hydroxyurea is the most effective for or preventing these vaso-occlusive episodes. Gene therapy is wonderful. It's an amazing science advancement, but from an impact perspective, we're talking about maybe treating 100 individuals a year. It's a very low impact, although at least from a numbers perspective.
That's mainly because the costs are prohibitive.
There's the cost is one aspect of this, but we've had, if you have a matched donor, you can have a bone marrow transplant, which is essentially curative as well. The gene therapies that are available today are very similar in how they're applied. You have a very harsh chemotherapy that primes your body so that you can do the gene therapy, which looks a lot like a bone marrow transplant. And it promises to be essentially curative, but you're spending several months in the hospital. The chemotherapies really, that carries a lot of risk with it as well. So, it's not just a matter of costs. It's also our hospital's fully equipped to be able to manage this. You can't really have a large number of people in the same hospital, sitting there for months while they're getting their therapy. So
that makes sense. And I'm sure, mentally and emotionally, there's very few things one would opt to sit in the hospital for several months over.
Yeah, and like some of the complications, it's infertility is certainly one of those. It's still, I guess, yet to be seen, but there's a potential risk for increasing the risk of leukemia down the road. Right now, that's not been shown in the gene therapies, but that's something that has been known simply just from the chemotherapy preparation.
By this point in our conversation, I was really feeling for the sickle cell patient community. I was ready to hear about the hopeful future John is cooking up at IHP Therapeutics. We're waiting for the solution. A good point to jump into what IHP is doing. So let's talk a little bit both about how IHP works. IHP 102 is one of the targets or products that you're working with. And in a scientific sense, what approach it's taking compared to the approaches that other drugs are taking out there or other drugs in development. Yeah, just what's the special sauce? How did you come to this point?
Yeah, so IHP 102. So first, from a treatment paradigm perspective, and I think this is important. We can come back to it later. But being able to have something that is self-administered at home while you're traveling, at summer camp, et cetera, something that circumvents the healthcare system, I think is really critical in the design of a therapy. The molecule IHP 102 that we are developing targets two things. One is we call the stickiness. So the occlusion of blood vessels. There are adhesion molecules that will capture red blood cells, platelets, neutrophils. These aggregates are what block the microvasculature that would feed downstream organs. And so IHP 102 interferes with those adhesion molecules and keeps the blood vessels open. The other thing that happens in a vaso-occlusive episode, there are more of the misshapen or sickle red blood cells, and those become fragile and rupture. And what you'll get is a situation of sterile inflammation. So when the red blood cells rupture, their contents actually do cause an inflammatory process. And if you have a little bit of the ischemia, that ischemic tissue can also be pro-inflammatory. And so there's a mechanism called complement, and we disrupt the complement pathway as well.
Okay. And was this molecule out there for a while? Like, there's always people looking for the next new drug to pounce on, and like generating so many new drugs computationally. How did IHP Therapeutics come upon it?
So it's a great question, and there's a couple of answers here. So there's an old molecule called heparin, and most people have heard of that. They understand it as an anticoagulant. What's interesting about this is that there are coagulopathies. So sometimes during a vaso-occlusive episode, people will have a heightened risk for blood clots. And so heparin had been used in some instances for that purpose. And in treating the coagulation issue, also discovered this is working to help reduce pain. And so there was a study in Saudi Arabia, for example, and then another one at Duke that was published that used a heparin molecule and found that there was actually a benefit. One of the problems, of course, is you don't necessarily want to anticoagulate somebody. So the molecule that we have designed, it builds from those findings and is a unique version of a heparin-like molecule. And so the underlying mechanism as well, one of those is a drug called crizanlizumab is now approved. There's another one in development that interferes with a adhesion molecule called P-selectin. And heparins are known to interfere with that, but heparins are also known to interfere with a number of other pro-inflammatory and complement cascades. And so just by tweaking that molecule or engineering it in certain ways, you can dial in the properties that you want and dial out the properties that you don't. And so that was the genesis of IHP 102.
And what were the 3 or 4 most important parameters you were kind of dialing for as you were experimenting with different versions of the molecule, maybe different formulations of the dose, that kind of thing?
Yeah, so the target activities were the first point. So we wanted to make sure that our P-selectin activity was as potent as we could have it, as well as complement, and then dialing out, making sure that there was limited anticoagulant activity. Having some anticoagulant activity could actually be beneficial, but if we're going to be applying this in the home setting, we want to make sure that we're still safe. So that simply from a biological perspective, those were some of the parameters that we optimized for. And then as far as formulations go, we want to make sure that this is something that could be administered in a way, so subcutaneous route. This could be not so dissimilar from insulin or GLP-1s that everybody is taking for weight loss. There's a lot of drugs that are administered by that route. Heparin is oftentimes administered by intravenous route, and that really requires the hospital setting. And in this case, we want to make sure that this is something that you can apply at home.
Because John brought up insulin, the affordability question came to mind. I wondered what he could control in the R&D process as a scientist to minimize its eventual cost to patients.
From what I understand of the company and like some of the accelerators you've been a part of, getting wide access and impact is really important to IHP Therapeutics. Is cost something you're able to think about or engineer for at all in the scientific room, or do those decisions happen in a separate part of the company?
Yeah.
What leverage do you have there, if any?
It's an excellent question. So absolutely, we do want to make sure that it's not just the 100,000 people in the United States, maybe about half that in Europe. That's typically where you make money off of a medication. The rest of the world is really what is afflicted the most with sickle cell disease. And so we absolutely want to make sure that this is something that everyone can access. That said, your question, I think, is it's the right one. How much do we incorporate cost into the science? And at this stage of development, you do try to make things as simple as possible, but designing drugs is not straightforward. And so it's hard to, kind of control for cost at this stage. What we do, though, in the long term, I think just simply because the drug class is not as challenging from a manufacturing perspective, I do anticipate that this is something that could be applied through all of the world.
In the last part of this conversation, we step away from the technical stuff. We dive into John's journey as Chief Scientific Officer, from wins and opportunities to difficult decisions, and hear how Stanford Biodesign changed his approach to science.
And just to get our hands on a little bit more of the science, since that's your area of work, have there been any particular experiments or pieces of data that you've worked on that were big highs for you or were really surprising? What have been the most rewarding parts of the scientific journey?
Yeah, so I think that you make baby steps along the way. You sort of set out, I have this thing in mind, but science is always one of those iterative processes, and it always challenges your thinking at every turn. And so This program has gone very well at each of those baby steps, but I would say one of the big inflections. I remember I was traveling, and one of our collaborators, he had been running a study. This is now published in Hematologica. But I was on an airplane about to head home, and I got an email from him. It said, "Your drug worked really well." This is just like, we got way better improvement in the vessel occlusions in this. There's a mouse model of sickle cell disease, and that was as much information as he sent in that email. And I just remember that flight was a fun one for me. I was quite happy to see that, and frankly, the data from those mouse studies really has been impactful. It translates well to the human condition, and we're still getting more data now. We have a couple of other collaborations, and the new data are all looking very promising. So we're excited about that. But I have to say that first one was probably the most fun.
Yeah, that makes sense. Along similar lines of just what it's like being in the chief scientific officer role, we're used to seeing a lot of interviews about the difficult decisions that a CEO or a CFO has to make, but I'm sure there's a lot of strategic planning and decision making in science. Like the process of science is like waking up each day and deciding which experiment to run. So maybe what's one of the hardest scientific decisions or experiments you've had to design in this role?
That's a tough question. I think when we work with collaborators, oftentimes you'll have different opinions on how you want to proceed, and I think sometimes there's a common ground that you reach. Generally speaking, I think we're pretty aligned with all of our collaborators, but there's only so much that you can do, and so oftentimes you'll hear from, whether it's an investor, a potential pharmaceutical partner, et cetera, I want to see these studies perform. And you're going to hear a lot of different opinions on those. And if we were a large organization, maybe we would do all of them. But as a small organization, you have to pick what is going to be the most valuable. And really, again, if you keep the patient, the individual as the North Star, I think you always work towards what experiments are really going to be most impactful in that regard. Now, some of those, it becomes irrelevant. It's sort of a biological question that needs to be answered anyway. But those are the kinds of things that I would say on a day in, day out basis, you do have that kind of in the front of your mind.
That makes sense. You were talking about keeping the patient in the center, and I can't help but thinking that probably comes from your Stanford Biodesign training somewhere or the other, because I also got to take a few courses with them. It's a totally different way of designing any type of solution. How has that training informed your work in this role and the way this therapeutic is being built?
Yeah, so the Stanford Biodesign program is really clinical needs driven. So we as scientists and engineers can oftentimes fall in love with some sort of technical solution before you realize, well, what problem am I actually solving for?
Everyone's been there, yeah.
I would say, yeah. And I absolutely have been guilty many times. But in Biodesign, the first several months of the program, all you are doing is writing down needs that you are seeing in the hospital setting. And this could be the operating room. This could be, I actually found many more needs following patients and their care providers in the office visits and so on. Really just learning, what is it that you are experiencing and what are the pain points for you. This is something that I think absolutely has driven our work here in sickle cell. If we were just thinking of this simply from a biological or a medical perspective, we might be looking in the hospital setting, for example, and say, well, you have a captive audience there. From a pricing perspective, maybe that's going to be better. But the sickle cell community has been saying for a very long time, this healthcare system is not working for me. I need something that empowers me, that allows me to travel, that does not subject me to that kind of treatment that I'm getting in the hospital setting. And so that really became integral in our development. And I think ultimately will hopefully lead to a very successful and impactful product of it in the end.
Does the fact that you're circumventing hospitals affect the development of your product in any way? It might hurt acceptability of the therapy in some way that it's circumventing hospitals and they kind of have a different interest there. How is that playing out, or is it more of a theoretical concern than a practical one?
I think when you talk to hematologists, especially those who specialize in sickle cell, there is absolutely no pushback whatsoever. There is sort of a clamoring for something like this. I think they know their patients very well. They realize what kind of impact this would have. It depends on from folks who are used to a typical clinical trial design. Doing something in the home setting can be challenging, and so we have to be extremely thoughtful in how we would do our clinical trial development. But I would say otherwise, there's not a pushback. It's not, I think hospitals are not a stakeholder that would be pushing against this either. I don't think that they're excited to, get paid more for having people with sickle cell and taking up a bed. This is absolutely something that most stakeholders are aligned.
That makes sense. That's great to hear. It sounds like it's going to be very impactful for patients when it's out there. I'm also curious, you were talking about how IHP 102 as a molecule came from heparin, and in academia, it's usually the case that a molecule is discovered and then people look for uses for that molecule. And that's the total opposite of the ideal pattern that kind of biodesign asks you to follow. And probably in practice, it's a mix, right? There's some amount of, you have new technical ability you want to do something with, and some amount of looking for a need fit. How was that for IHP? Was it closer to the biodesign pathway, or how academia goes about these things?
Yeah, great question. So, and there's maybe a mixed answer on that. I would say, in part, the biodesign process says do not start with a solution, start with a problem, solve for that problem. Sometimes, though, it marries well with what your background information. So I come from a background in glycobiology. I've been studying heparins for a very long time, and understood that boring old anticoagulant drug actually has a whole lot of other activities, and maybe there's something that we could exploit here, with certain designs. So I think that those two kind of came together, really understanding the problems in sickle cell and understanding the pathomechanisms, what other drugs have worked in the past. What we also did, though, was look at how many other ways could you approach this? Are there other drugs that could have solved the same problem in a better way? We didn't have the hammer and we're just looking for nails. We said, what is actually the best tool for that? And can we, if it doesn't exist today, is there something that we can do to design the best tool? And that's ultimately what led to IHP 102.
That's really amazing. I feel like I'm about to start my PhD in August, and I don't think that's the way it happens in most places, even in really great institutions that do a lot of good work. So,
well, I think always coming into it with what problem am I solving for is going to position you for better success. I think, if you come into this where I'm just driven by interesting science that doesn't necessarily have the end application, certainly there are examples of where basic science purely for the scientific discovery has given us great advances. But I tend to think that really, if you want to have more immediate impact, making sure that you're solving for a problem. And then, Biodesign is at Stanford does now have a biopharma effort. And really in this effort, when you do understand pathomechanisms and so on, you can start to say, well, we have advanced far enough along in biopharma to where I can generate a molecule that, say, like an siRNA, is going to have a very long durability if I know which genes I need to shut down. That's going to be a better suited solution for certain conditions. Maybe a small molecule is better for this one. And, there are ways of designing those as well. But, I do think that kind of keeping the problem at front and center and really understand the need you're solving for is going to make a greater impact.
That makes a lot of sense. Yeah. And especially, I guess doing biodesign after having a lot of technical experience positions you very uniquely to use that knowledge in a way that a first-year undergrad student doing biodesign wouldn't be positioned.
Yeah. Yeah, I laugh. I think about myself pre-Biodesign and kind of the more of a mad scientist saying, "Oh wow, what did, this is really cool," and then what can we do with it? And seeing myself today, it has absolutely changed my perspective and really how I focus my energy.
Yeah, I'm looking to find that balance. I think there's pockets of academia where doing science for science sake is almost celebrated. And I think personally I'm differently driven, but I'll have to figure out that balance.
Yeah, yeah. No, I'd say in the medical field, having a patient, somebody who is suffering as kind of your guiding light, that absolutely is motivating. And so, science for the sake of science, I think, can be motivating too. But there's a different level of motivation, I think, when you think about the potential to alleviate the
That
suffering.
makes sense. We'll start winding up. The process of developing a drug like this and getting it out there takes years. So, there's nothing in the market right now, but I do want to celebrate, like, what are the milestones looking out to the next few years of development? Is there an opportunity or risk that you see being really significant in the future?
Yeah. So the other thing that we have been very fortunate, the government agencies, as I mentioned, have a significant interest in helping to develop new therapies for sickle cell. And so we have been fortunate to have significant government funding to support the development of IHP 1 and 2. Ahead of us, we obviously have a lot of work to do at, making the molecule, running through the safety studies, et cetera. We're kind of in the beginning phases of that right now before we get into the first patient. Designing the clinical trial, as I mentioned, is something that we're going to have to put a lot of thought and energy into to make sure that is run well and really can demonstrate the benefit of the drug. And, I would say a typical small company like ours will oftentimes, a typical path would be to find partners, once you get into the clinical trial phase and to help, support those efforts. So we're looking at everything ahead of us. I think there are plenty of risks. As you said, it's a long process. This is a marathon and we're ready for it. Like I said, as long as you stay motivated, and there's plenty to keep yourself motivated in this one.
That's amazing to hear. And all the best with the years ahead of the journey. So it's been great talking to you, John. I usually like to end with a small personal question. What's something simple you do every day, every week for your own health or the health of your community? And then we'll close it out there.
So, what do I do every day? I am trying to, at this point, exercise, I realize, is one of the healthiest things we can do for ourselves. That is something that, even if it's just doesn't have to be a long run. It could be just, anything every single day. I try to do that. And then I love working with fellow scientists. So I am a mentor to a number of young budding scientists and some startup companies. So those are things that really energize me as well.
Yeah, on the exercise point, I saw on social media a study circulating recently that said dance is more effective than SSRIs for treating depression. And I'm not going to put my weight behind that because it was on the internet, but it's insane what can be fixed or what can go wrong if our bodies are not moving the way they're supposed to.
Yeah, I could, not having read any of the science behind that, I do kind of on the surface think that there could be something to that for sure.
Yeah. Thank you so much for taking out the time, John, and for your patience with all the technical difficulties. Really insightful conversation.
Well, I really appreciate you inviting me for this, and this has been fantastic. So I really appreciate it.
Thank you.
Thank you all for listening. If you like this episode, leave a rating and review wherever you get your podcast. Follow Cell Culture Community on Instagram, this podcast on Spotify or Apple Podcasts, or just stay in touch with me for more conversations on how science and technology can transform our health. Till then, and for the last time this season, breathe deep, stretch something that feels stiff, and look around. Notice something curious, delightful, or in need of care. We are gifted with so many chances to love and wonder every day.